In the overwhelming majority of cases, existing medical simulation systems have been developed on the basis of mathematical modeling methods, formalized as complex sets of equations describing processes of respiration, hemodynamics, cardiac activity, muscular work, etc. Such modeling results converted into physical signals drive, for instance, widely known mannequin-type simulation systems. It is quite obvious that any medical simulator built on such models, even with the best engineering solutions provided, has nothing to do with the real structure and processes in a living organism. A condition of an organism can be described solely by a totality of interrelated conditions of separate organs and systems, which makes a system approach the only trustworthy method to model an organism. The use of technologies described above excludes this approach in principle. Such models may appear to be relatively trustworthy, positive exclusively in description of isolated processes in a particular organ or system, but it is merely impossible to bind them with a multitude of other concurrent processes. Despite some interesting results obtained in biology and medical science by means of mathematical modeling methods, development of such has no prospects. They have reached the deadlock. This problem is a long-standing one. Back in the 80s of the 20th century, still in the USSR, at the joint meeting of mathematical and biological sections of the Academy of Sciences, biologists complained of poor performance of mathematical models developed to describe biological processes. Academician Gelfand, a world-famous scientist then, commented that the language of mathematics was meant to describe physical and technical processes with little use for biology. He who wants to model biological processes should develop a special language for description. We present PureMedSim, a principally new technology of modeling as close as possible to real processes in human body and built according to the pattern from down to up: cells, organs, systems, and organism. A virtual organism arranged thereby consists of cells, tissues, vessels, organs filled with water and all necessary chemical substances. To describe the organs and systems of the virtual organism, we engage more than 130,000 biophysical, physiological, and biochemical indices, plus millions of structural units. Our software code is, by its nature, the new language of description the great mathematician meant. Computational methods, algorithms, presentation, and arrangement of data arrays, virtual movement instruments, bringing together all stages of the technological process as a code represent the absolute know-how. The following animation shows the stages of the technological process. Here is the organism and weight percentage of various organs and tissues. Modeling process will be shown by the example of muscle tissue. Other tissues and organs are modeled alike. The first stage features formation of extracellular environment. The software code creates corresponding data classes and arrays. Then cells of the tissue or organ are being filled with necessary chemical substances. The next stage is a key one in our technology. All cells' biochemical transformations are being modeled within. Then the tissues and organs as such are being formed. The necessary cell count is determined, and cells are distributed throughout extracellular environment and stroma. In the same way, the vascular system is built. At first, blood plasma and then blood cells are being formed. Specific biochemical transformations in blood cells are being modeled. Then virtual mechanisms of blood flow in the vascular system are being developed. After that, the vascular system is being built into corresponding tissues and organs, and metabolic processes are being modeled. In the final stage, the organs are being built into respective systems, as shown, for instance, for the system of hemodynamics. A number of products have been developed on the basis of this technology. The pilot one is the next generation medical simulator PureMedSim Live, an unparalleled effort as a large-scale living organisms model comprising 16 organs and systems. Interrelated biophysical, physiological, and biochemical processes concurrently proceed in the simulator in the real-time mode. Each subsystem in the simulator is represented by a set of certain quantitative indices describing particular functions of the organisms, organs, and systems. Average values of all indices for all subsystems have been set in the system by default. Those are well-known values provided in medical reference literature and on websites. All the formulas applied are also of common knowledge and are described in textbooks on physiology, biochemistry, biophysics, molecular biology, chemistry. A practical solution of the simulator is presented as a 3-monitor system, enabling an operator to concurrently observe functioning of several organs and systems at the same time. A user can choose a desired level of detailing. Such may be an organism as a whole, an organ or a system, and a cellular level. A 3-monitor solution makes it possible to choose all the levels simultaneously and for different organs and systems. Technical limits of this presentation reduce demonstration of the simulator's work to series of pictures on the screen, the so-called single-monitor version. Having started the simulator, we see the following. The center of the screen is occupied by a screen simulator, a virtual organism. It is able to display the main externally observable processes: respiration, heartbeats, winking, changes in skin color, weight loss. Biophysical, physiological, and biochemical processes proceed in the virtual organism and are displayed in the real-time mode as vitals in the left part of the main menu. Here the monitor readings are enlarged. The fundamental difference of this simulator from the rest of existing medical simulation systems is that all these readings have not been obtained with the help of mathematical packages, nor have they been drawn in a graphics editor or scanned from some real sources, nor are they animated fragments. These signals are being generated by this system at work. Let us demonstrate some operation modes of this simulator. We will take as an example functioning of the cardiovascular module on all levels from the cellular one. This is a video of a virtual cardiac conduction system sinus node cell working. In the real-time mode, the membrane channels open and close. Ion concentrations on the two sides of the membrane alter. Electric potentials change. ATP-driven potassium-sodium pumps work. As a result, an action biopotential is generated in complete correspondence to normal action potentials of real sinus node cells in a real organism. Action potential duration and form depend on a part of the conduction path or myocardium's working sections. This is action biopotential of cells in the bundle of Wenkenbach. This is action by potential of cells in the bundle of Bachmann. This is action by potential of atrial cardiomyocytes. This is action by potential of cells in the bundle of Thorel. This is action by potential of cells in the AV node. This is action by potential of cells in the bundle of His. This is action by potential of cells in the bundle of His branches. This is action by potential of left ventricular cardiomyocytes. Please note that a contraction occurs in cardiomyocytes of atria and ventricles as well. As in real cells, a muscle contraction mechanism is connected with a power stroke of actin and myosin filaments. A user can interactively alter membrane channels permeability, thus changing action by potential duration and form. Here is a normal action potential of sinus node cells. Under certain changes in membrane permeability, a pulse rate may increase, for instance, by 5 times. In our software code, all virtual cells are brought together into a unified structure, reflecting a virtual conduction path, and are arranged in the virtual 3D space. It allows to obtain a general all-out picture of action by potential generation by cells of various conduction path sections along the wave of excitation. These impulses fully correspond to those demonstrated above, but shown in a more condensed scale. The impulses propagate asynchronously. Propagation time in each section corresponds to real-time scale. Spatial summation of these impulses, with taking into account the chest's geometry, leads to ECG generation from heart cells microcurrents. The ECG is being generated in 12 standard and chest leads. Any lead can be zoomed in. The system has been built so that a possibility of interactive studies of mechanisms of various disorders in organs and systems functioning is provided. This can be demonstrated with single extrasystole occurrence taken as an example. Yellow squares show locations in each part of the conduction path, acting upon which one can study in real-time mode mechanisms of single extrasystoles initiation from various sections of the heart. Let us choose the AV node, for instance. Acting upon the node we have chosen by a 10 mV impulse within a certain time interval, we obtain as a result a premature heartbeat. The next video features the system of hemodynamics. In the hemodynamics mode, one can observe an outline of blood flow through the virtual organism's vessels in systemic, pulmonary, and coronary circulation. It is a complete, all-out, detailed blood circulation outline. It demonstrates all directions of the blood flow with taking into consideration paired organs and their blood filling patterns. Each route is marked by corresponding indices. This outline fully corresponds to real blood circulation in a human organism in recumbency. Computational algorithms are in complete agreement with the theory of blood circulation physiology. The interactive mode allows to narrow arterioles lumen down to complete blood flow stop in the organ's vascular system and observe changes in corresponding indices on the general blood circulation layout. This will be demonstrated with the left kidney taken as an example. The layout shows how blood flow gets redistributed and related indices change. The next video shows coronary system. This mode demonstrates how oxygen is carried through coronary vessels to respective sections of the heart. Mapping of coronary arteries with blood pressure and volume indices in each branch is also provided. This mode makes it possible to model vascular permeability pathologies in any part of the coronary system. That will be shown later in scenarios of pathological processes. By means of this technology, with related cellular mechanisms engaged, and in full correspondence to real physiological processes in an organism, there have been built models of respiration, gas exchange, gastrointestinal tract, excretory system, etc. The following is a brief survey of such systems' functions. Respiratory system and gas exchange. The lungs breathe in the real-time mode in complete compliance with real respiratory mechanics. Negative pressure is being formed in pleural cavity due to contraction of diaphragm muscles. ATP energy is consumed in the process. Also in the real-time mode, blood is passing from the heart to the lungs, gets oxygenated in alveoli, and returns to the heart. Gas composition can be monitored along the inhale-exhale chain. The interactive mode makes it possible to manually regulate respiration rate and respiratory depth, as well as alter patency of bronchi. The following dynamic diagrams provide the possibility to observe in real-time mode gas exchange in various tissues and organs. Let us take liver as an example. Oxygen is shown in blue and carbon dioxide is shown in yellow. The following is a brief demonstration of excretory system at work. This video shows water and salt metabolism. Red color and corresponding indices stand for content of water in blood, cyan for water in each organ's extracellular environment, blue for intracellular water. Dynamic fluctuations of quantitative indices for various substances of liquid mediums are also provided herein. Water and salt homeostasis is provided by a nephron with processes of filtration and reabsorption. For normal functioning of the virtual organism, as well as for a real one, it is necessary to provide inflow of fats, proteins, and carbohydrates carried in with foodstuff. The system's database features various foodstuffs with information concerning their nutritive and food value. Digestion proceeds in the gastrointestinal tract with the help of respective enzymes. It is commonly known that an organism's vital activities are based upon various biochemical processes. In all existing medical simulation systems, the module of biochemical and enzymatic transformations is not represented at all, with just a few constants and variables in a model's equations instead. Here, in real-time mode, we demonstrate all series of biochemical transformations of various ingredients in each particular organ and subsequent biochemical transformations of substrates obtained, including the Krebs cycle and ornithine cycle, as well as energy production processes in particular organs' cells and exit of irreversible decay products. Biochemical processes presented provide generation of energy the organism needs to perform its vital activities. The most interesting facility of the system is that dealing with clinical scenarios of pathological processes. The modeling technology makes it possible to create scenarios in such a great detail that results obtained remain unattainable for other simulation systems. Each scenario is a specially designed form containing description of a pathology with a down-to-cellular level refinement, and features interactive windows to operate corresponding scenario mechanisms. A description of all possible treatment methods is also provided therein, and the system's database comprises necessary drugs and instrument means. Let us take ventricular paroxysmal tachycardia as an example. The scenario window provides a brief description of the pathology. In this particular case, the disorder is caused by a locus of pathology in the left ventricle. The locus consists of 4 zones with slow depolarization where persistent cyclic impulses are generated, which leads to higher ventricular contraction rate. The scenarios have been designed to enable a user to interactively interfere in the pathology course. For instance, one may regulate action biopotentials in the locus zones. Finally, the scenario provides a dynamic interactive graph showing the propagation of the excitation wave through the pathways of the heart under this pathology. Yellow squares stand for unexcited cells, red squares for excited ones, and pale red squares for those in the refractory phase. The group of pathological cells is shown in pink, green, blue, and cyan. Digits within squares show time elapsed since excitation emerged in the sinus node in milliseconds. A user can manually slow these processes down to step-by-step performance. Corresponding changes can be observed in the ECG and the graphs of propagation of biopotentials through the pathways of the heart. There are 2 ways to deal with this pathology: drug and electropulse treatment. We will show both of them. Let us begin with medication. The system's database contains corresponding drugs subdivided into groups. To obtain a quick response, we will inject lidocaine. In 20–25 seconds, the rate is restored. A user can, at the same time, observe this drug's pharmacokinetics and follow its uptake in extracellular and intracellular environment, gluing to proteins and albumin absorption. Another option is cardioversion of defibrillator. Let us begin with a minimum charge of 150 joules. As we can see, no positive result has been obtained. The charge goes up. It is 250 joules now. Now the rate is being restored. Here is another scenario. Phrenic myocardial infarction model caused by blood clot in the right coronary artery. At first, in the ECG, we observe a downshift of ST segment, which is characteristic of endocardium hypoxia. Subsequent elevation of ST segment signals about a damaged zone formation, shown in darker shade here. The corresponding changes in hemodynamics and gas exchange will take place, which can also be observed by a user. A timely dealing with this abnormality restores blood flow and the ECG gets back to normal waveforms. We have briefly demonstrated our virtual organism project. The basic model has been engaged in practical use already and features 30 clinical scenarios, mainly within cardiovascular system. Further on, the project will be developed in two directions, enhancing financial potential and adding new scenarios. It is important to understand that this process is infinite, for studies of a human organism are inexhaustible. In half a year, the basic model will be enhanced by the nervous system and elements of cerebrum physiology connected to already existing systems, which will be yet another unparalleled achievement. Scenarios of hemorrhages and pulmonary embolism will also be built in. Then groups of our experts will proceed with making new pathology scenarios for various organs: brain, kidneys, liver, lungs, gastrointestinal tract. According to our calculations, we're quite capable of developing up to 40–50 scenarios annually. We stress yet once again that the model we have just demonstrated will remain the basic one. The system may soon become a new standard in teaching medicine. Such a decision has already been made in Ukraine. The general idea is that any first-year student of medicine will begin studying theoretical disciplines with the help of this simulator, never to part with it throughout his professional life, for the system will be constantly updated with new functions and scenarios. In time, it will become important for clinical practice as well, for the system will help a clinician to quickly determine the cause and mechanism of a disease and provide proper and timely treatment. The system may also be widely applied both in scientific studies in various fields of medical theory and in practical research work, in pharmaceutics, for instance.